So thank you, Nizar, for the introduction, and before that to you and your colleagues for the invitation to come to talk at the 18th Saudi Society for Medical Microbiology and Infectious Diseases meeting. Now, when Ghassan contacted me and asked me if I would be willing to come and speak in this conference and give a plenary, I said, yes, of course. It's always a pleasure to be in Jeddah. And he said, what would you like to speak on? And then we had some discussion. And we agreed. We agreed on the title. But what we didn't agree on, to be honest with you, and I found out about later, was A, that he was going to put me straight after lunch, and B, that he was going to expect me to speak for 1 hour. I'm too old to speak for an hour. So I'm glad in a way that we're starting 10 minutes late. So what I intend to talk about are clinical trials and clinical trials only that changed my practice. Of course, these are trials that may well have not made any difference to your practice, and you may agree or disagree with what I've decided to change in my practice based on those clinical trials. That's all up to you. But a lot of the story is about the timeline. And the timeline is very important because when I started doing ID, Google was pretty new. It had existed for only a few months when I started doing ID. Arsenal, who are still my favorite football team in the English Premier League, were doing incredibly well. I think they're doing better this season than the season before, but we've had better days. The Euro didn't even exist. It started about 2 months after I became an ID, what's the equivalent what you would call a fellow. And certainly Facebook didn't exist for at least another 4 years. So where I was when I started doing ID is quite different to what many of you in this room, not everyone, but many of you in this room were at the same time. So some of what I will describe is different to what you've experienced as you joined your journeys. So when I joined ID, the biggest issue of the day was HIV infection. It was the late 90s, and it was literally equivalent to a very miserable life, leading invariably to a premature death. And it wasn't an easy death. It was always associated with a lot of pains and aches. The fact that we eventually managed to find some antiretrovirals that can help some patients was great news. But as I was starting doing ID, the treatments that we were using were largely of very limited effectiveness. If a patient is able to take them, and if is a very big word, we heard earlier that Moyed never prescribed TDF. Now he would be shocked to learn that I actually described zalcytabine and zidovudine and some of the ancient agents. But those agents were good at suppressing the virus, but only for a very short while. Now, what changed the world is a trial like this one. This was the spark that changed everything. This was a randomized controlled trial of a large number of patients who were obviously HIV infected, and they were randomized to receive 2 NRTIs. They were stavudine and lamivudine versus adding a protease inhibitor. This is what happened to the group who received 2 NRTIs, so the viral load decreased, but it rebounded very quickly, as we all know, as resistance emerges. But this is what happened to the arm who received nilfinavir, a protease inhibitor that, again, many of you would not have prescribed. Now, when you add nilfinavir, when you added nilfinavir in this clinical trial, what you got is viral load suppression, but more importantly, sustained viral load suppression. And with this sustained viral load suppression, you saw the next thing, which in my days was unthinkable, that you would actually see a reversal of the CD4 count decline. So as you've managed to suppress the viral load and keep it suppressed long enough, you can start to see immune reconstitution. And patients who you saw with CD4 counts that are 10 and 20, you can see them after a year or two with CD4 counts of 200. And that was absolutely unthinkable that this was achievable. But we were using those protease inhibitors. They immediately changed our practice. Now, I could say that I am one of people who are very fortunate to live through such a breakthrough. In the first year, just 1 year after introducing triple therapy based on protease inhibitors, mortality associated with HIV decreased by 90%. Now, if you were a physician in the 1940s and you witness what penicillin did to infections, you would be as thrilled as those like me were around those days, seeing what happened with the introduction of the PIs. So this was a practice-changing trial that led to a change, a fundamental change in how we look after patients. But once we used protease inhibitors, we got into the next stage. And that next stage was one where patients were surviving, they were living longer, but they were also experiencing extremes of toxicity. Some of it was just terribly intolerable medication, constant nausea, very severe vomiting, diarrhea, abdominal pain, and it just didn't feel right. But some of it was also metabolic, and some people died prematurely because of complications related to the medication, things like pancreatitis, hepatitis, and obviously cardiovascular events. So the next thing that we needed was to try to find better drugs. So the next trial that changed what we did, changed what I did, just to stick with the title of the talk, was a study that's known as 006. And if you're my generation, you don't need anything to be introduced to 006. It's the equivalent of naming any famous or celebrity that you know. 006 was the gateway for introducing non-nucleoside reverse transcriptase inhibitors into broad or wide clinical practice. It introduced efavirenz, but it did two things that are very important for me now as I reflect and share with you that you realize. The first is that we know now, we knew then, that PIs work very well, but they're horrible. What we wanted is something that can replace them. What we got is something that was even better. It didn't just replace the effectiveness of PIs, it actually superseded that. So if you look at the results of the trial 006, you could see that efavirenz-based therapy was actually better than PIs. And that meant an instant change in practice into NNRTI-based therapy wherever possible, away from protease inhibitors. This was efavirenz in combination with Combivir. Again, perhaps an unfamiliar name to you, but not for many in the front row. Combivir is the combined pill that associates zidovudine with lamivudine. But then the real leap in what I did and many people did was the introduction of TDF, a drug that is now not even known well enough. The introduction of TDF with FTC and then demonstrating that it works in study 934 when combined with efavirenz was a complete leap forward because it did two things again at the same time. It introduced a better NRTI, that is tenofovir, and at the same time brought this. This is the equivalent of Steve Jobs walking up and down this podium showing you the iPhone, if you've never seen that video. Again, I show my age when I talk about that video, but it was quite impressive, and you haven't seen it yet, YouTube it. I think you'll love it, because he walks up and down the stage, and then he pulls off his, well, would later become known as the iPhone, and he says, I want an electronic organizer, and I want an electronic email thing, and I want something to call, and I've got it all in one thing. Well, the equivalent of that was a triplet, because that study, 936, allowed efavirenz with tenofovir FTC to become standard of care. And then they were, when they were all combined into a single pill, this was unbelievable, because everything before that was based on a large number of pills that were not easy to take. And it wasn't the only evolution development, but those stand out. The next thing after the introduction of NNRTIs, the next big trial in HIV that changed my practice, changed a lot of people's practice, was the introduction of integrase inhibitors. Now, the first integrase inhibitor to make it through was raltegravir, and the randomized trial that demonstrated its effectiveness was the STARMARK. And again, STARMARK is very well known. So what happened in STARMARK was patients with HIV who were naive to treatment were randomized to receive raltegravir-based treatment, this new, shiny class of antiretrovirals, with our gold standard of the day, efavirenz. And what it showed is two things. A, it was non-inferior, which is fantastic. But also, it showed that if you were to use integrase inhibitors, you can suppress the virus even quicker. And you can get that accelerated viral suppression within a much shorter space of time. Now, the other thing that you could see now as you look at the STAR mark results is that the proportion of patients who were still suppressed a year after treatment has changed. So the earlier studies I showed you, it was like 50 to 60% and then 70%, and now you're pushing it. And after a year of treatment, you have more than 80% suppressed on treatment. This particular study went on to have follow-up for 5 years. And at 5 years, it was obvious that raltegravir was superior to efavirenz. So this was the next move forward. This was the next trial that changed my practice, one that introduced integrase inhibitors into practice. And obviously, other integrase inhibitors followed thereafter. Now, if you start where I started speaking about a disease that was fatal and also causes a lot of horrible complications, and then you have treatment that is associated in itself with lots of horrible complications, the sensible thing to do would be not to give medication to those patients unless they desperately need it. So there was a time when we did not treat everyone who was diagnosed with HIV. There was a time when the sensible thing was to diagnose someone and say, well, let's wait until your CD4 count is low enough. And again, many people in this room would remember those days. So we waited, believe it or not, but that's what we did because that was the right thing to do at the time, until the CD4 count was less than 350. And the reason we chose 350 is that we didn't want them to get down to 200. So 350 is a mark that they're approaching that magic number of 200 or the worrying number of 200. So we had to wait. But as time moved on and the post-STAR mark study that I showed you now, you have better treatments that are far more effective and far less toxic. So the proper thing to do is to go back to the drawing board and ask, is it still appropriate and reasonable to delay potentially useful treatment? And that's where the STAR study comes in. This was an absolute practice changer because what it did, it is randomized patients who had CD4 counts of above 500. And I'll have to confess with you that when I first heard that there was a trial that is intending to recruit patients with CD4 counts of above 100, I thought, are they crazy? Why would they want to do that? Why would you want to recruit someone who has CD4 counts of above 500 and give them antiretroviral therapy? But the results of the study showed something that's completely different. So here you have patients who had CD4 counts of above 500, and they were randomized to either starting treatment straight away or not. And the first thing you see in those two graphs is those who started, obviously, achieved suppressed viral load and improvement in CD4 counts. But who cared? They already have CD4 counts of above 500. Well, this is the second part that changed our practice, because not only those numbers were better, those patients who started early were less likely to experience significant events. And by significant events, I mean AIDS-defining illnesses and death. So if you were to treat people while their CD4 count is in the healthy bracket, it still actually works and it still saves lives. So STAR changed our practice. It's not just that we have better antiretrovirals, we were now using them much, much earlier. So within an incredibly short period of time, if you want to take that perspective, we went from a point where treatment for HIV meant taking a very large number of pills, but you delay this as long as possible because they're not easy to take. And we went over to a place where we can treat HIV very effectively with a single pill and with very, very minimal adverse events. And if that doesn't change anyone's practice, then obviously nothing will. The next part of my career, and it's not a short career, believe me, was the second concern. The first thing that hit me when I walked into ID was HIV. It was the biggest thing. But the next thing was chronic hepatitis C. Chronic hepatitis C, as you all know, is a chronic infection that just doesn't go away. It's not like hepatitis B if you acquire it as an adult. So individuals who became infected with hepatitis B at any point in their lives are very, very likely to remain chronically infected. And then a proportion of them, as you all know, develop progressive liver disease, cirrhosis, and potentially cancer. The first thing that happened to try to treat hepatitis C was the introduction of interferon, conventional interferon, that had to be taken 3 times a week. And then the biggish breakthrough was the introduction of ribavirin. So when I walked into ID, the standard of the day to treat chronic hepatitis C infection was to give conventional interferon 3 times a week plus oral ribavirin. Now, again, I'm quite sure very few of you have prescribed interferon, at least in dose era. To give interferon is the equivalent of giving you influenza, because it's an immune modulator. And what it does is it stimulates all your immune system to try to fight off the virus. So if I give this to you 3 times a week, I'm practically giving you flu 3 times a week. And actually, I want you to do this for the next 6 months. And depending on your genotype, I might actually want you to do it for the next year. That obviously wasn't easy. The terrible thing was, it wasn't even effective. We achieved sustained virological responses in less than 20% of patients who we treated. So you know that 4 out of every 5 patients you treat are not even going to respond. So the most exciting piece of news I received when I started doing ID was the introduction of pegylated interferon. So what we have now is a molecule that is attached to interferon, and that allows less frequent administration. So instead of giving it 3 times a week, you give it only once a week. And then you wouldn't believe the excitement that we have. That now we can give interferon, we can give you influenza, not 3 times a week, but we will do it just once a week. So now, instead of having flu all the time, you have it all the time, because the pharmacokinetics is the same. So you feel tired and achy, and you have headache, and you're off your food, and you can't tolerate life, and you're expected to do that again for 6 months or 12 months, depending on your genotype. So what happened after that? And by the way, the SVR rates, the sustained virological rates, were somewhere between 30 to 40, 50% if you're very lucky. The breakthrough was the introduction of antivirals, those that inhibited protease inhibitors belonging to the non-structural protein 3. This is something that I actually witnessed, and I'm very happy I did, because it happened during my times. So suddenly, we have oral antiviral agents, tilaprevir, boceprevir. Again, not many of you would have used. But I just want to show you what happened at the time when those drugs became suddenly available. So they were tested in those clinical trials that I wanted to share with you. SPRINT2 and ADVANCE, they used these 2 different molecules with different background, interferons and ribavirin. But the point is, those 2 trials were in patients who had never been treated for hepatitis C before. And what they included is randomizing patients to the standard of care, pegylated interferon ribavirin, or slightly different regimens. And this is the first time we start to see these numbers. So as you can see, in those who were receiving our standard treatments, SVR rates are as expected, around 40%. But it was inconceivable that we would see treatments for hepatitis C that would reach something like 70% and 75% SVR in genotype 1. And even better, those 2 trials that tested the same products in treatment-experienced patients, so those who have not tolerated previous treatments or failed them, again showed impressive rates of SVR. So this was a practice-changing finding. So suddenly now, we're using boceprevir, tilaprevir on top of pegylated interferon and ribavirin. But this wasn't the last breakthrough. The real breakthrough in hepatitis C happened when sofosbuvir with velpatasvir were introduced, because these drugs work on all genotypes. And by the time they were tested in a trial, it wasn't even necessary or required to have a comparator arm. You could just give placebo. And these are the results of the Astral trial that changed my practice, probably changed everyone else's practice. Because look at the SVR rates. It's 99%. And just for good measure, the placebo arm had no one, obviously, who developed SVR. And the beautiful thing is that this comes once daily, and you take it orally, so there's no horrible interferon involved, and you only need it for 3 weeks. And you know what? This is the size of the pill. Now, who wouldn't change their practice if you had this size of a pill that gave you 99% SVR in the hardest to treat subset of patients with chronic hepatitis C? And obviously, as you all know from there on, lots of other oral antivirals have changed. I started at a time when hepatitis C was a nightmare. Treatment was even more a nightmare than the complications of hepatitis C itself. And I call it interferon the therapy, because that's what it did to patients. And if you've seen patients who've taken interferon, you know what I mean. And all that comes with little success. And obviously, we moved on now to a single pill that achieves SVR in almost all patients, And that's fantastic. The third issue of the day, still is somewhat the issue of the day, is obviously tuberculosis. Now, tuberculosis, the first ever antimicrobial was streptomycin. And it was obviously used for TB. But then a good part of the 1960s and the early 1970s went into trying to develop treatments that could be used to cure tuberculosis in a shorter time. So the standard care for tuberculosis in the early 70s was to give treatments that usually lasted for 18 months to 2 years. Now, it's common knowledge for us. It was even common knowledge for me when I joined ID that rifampicin changed the face of treatment for tuberculosis, and it became a short course, 6 months. Short compared with 18. Obviously, 6 is not necessarily short if you compare it with a much shorter time. And that went smoothly and nicely and cured immeasurable numbers of individuals from tuberculosis all around the world. And that remained the case until we started to see resistance to rifampicin and resistance to isoniazid, or resistance to both, multi-drug resistant tuberculosis. And that took us back to square one. So when I started doing ID 25 years ago, minus 2 months, the standard treatment for people who have drug-resistant tuberculosis consisted of no less than 6 to 7 drugs. At least one was an injectable, usually an aminoglycoside. And you're expected to give that aminoglycoside typically for 6 months. And then you combine that with a number of oral agents, none of them is easy to take. If you treated multi-drug resistant tuberculosis the time that I was treating them, those days, actually for a long time after that, you would remember patients who went deaf, irreversibly deaf. You will remember patients who needed dialysis eventually because of irreversible renal failure. You will remember patients who experienced horrible, painful peripheral neuropathy because, again, of the side effects of the treatment. And it was all based on a very basic principle. You needed an injectable and you needed many, many medications. This mind shift happened with the introduction of the so-called Bangladeshi regimen, because Bangladeshi regimen refers to the first so-called short regimen to treat multi-drug resistant tuberculosis. And what they did in a series of observational studies and then eventually the STREAM trial, which is the one I'm sharing with you, is they used this regimen, which constituted an injectable with at least 6 other drugs for pulmonary TB, MDR TB. And what they showed is that when you compared that with the standard course, which was 2 years, it did well. And that is fantastic news. So suddenly, instead of having difficult to take medication for 2 years, maybe you can do it only for 9 months. STREAM. But things got even better. Better in a way that I would not in my dreams have expected or predicted. Suddenly, not suddenly actually, this happened over 2 decades, but suddenly, we have 3 new agents. Bedequiline, which was developed specifically as an anti-tuberculous agent, an antimicrobacterial agent, britomanid, and separately linezolid, which, as you all know, was developed to treat gram-positive infections. So we now have those 3 agents with very useful activity against tuberculosis. Now, what if we use them in one regimen, and what if we call that regimen B-PAL, which makes sense? Well, this is the first trial to see what happens if you use those 3 drugs for drug-resistant tuberculosis. Now, the piece of amazing news here is not just that these are 3 drugs. The course is only 6 months, and all 3 of them are given orally. So now in this trial, the NICS-TB, a regimen based on B-PAL was compared with standard treatment. And the response rates were incredibly good, way too good, almost too good to believe, because the previous regimens that were very long were also not associated with impressive success, typically around 70, 75%. Now you're pushing it all the way to 90% in 6 months with a drug regimen that is all oral. And unfortunately, it was associated with lots of side effects. Now, the side effects, in particular peripheral neuropathy, was largely driven by linezolid. So we had a second trial, the NICS-Z, that tested the dose of linezolid, and it was possible then to optimize that down to 600. It wasn't the only clinical trial. There's a second one, TB Practical, which published more recently. Again, B-PAL, but this time with moxifloxacin, and again for MDR-TB, and again for 6 months. So where are we today now? These are the adverse events of B-PAL. Unbelievable. 3% hepatotoxicity, and 1% QT prolongation. So we came from a point in the 40s and 50s where tuberculosis was not treated, to a point in the mid-70s where it became relatively straightforward to treat. And then we declined again and went into multi-drug resistant tuberculosis. And again, we're using treatments that were highly toxic and not so effective. And somehow we managed to turn the tide again. And we again have regimens now that can be given orally, all oral, for 6 months to achieve very high cure rates of multi-drug resistant and even extremely drug resistant tuberculosis. So going all the way from the injectables and their companions to PPAP, relative simple treatment that is very, very effective. Now, if you and I, well, if you were with me those days, and I was introducing to you a disease that is relatively novel anyway, HIV, it's not very difficult for any of us to change their practice when new data come up, because there are no dogmas to hold on to. Anything that we learn together. So it's not particularly impressive that I change my practice whenever there's an obvious reason to change your practice. But what if you were somehow being passed over a dogma that has been hanging around for the last so many decades? And suddenly now you're asking yourself, do I really want to change my practice? Or do you want to change your practice? That's not the same. And there is no old foe in infectious diseases. There's no older foe than Staph aureus bacteremia or Staph aureus infections in general. And we had a wonderful session on this morning by Ibrahim Mahmoud. So one of the dogmas around treating Staph aureus bacteremia is, do you need one drug or do you need more? And in my days, in my earlier days at least, a lot of people loved giving a second drug. And that second drug typically was rifampicin, because it worked in vitro and it must work in vivo, and because everyone else did it. But it wasn't tested in what you would call a proper, robust, randomized trial until ARREST came on. So in this trial, patients with Staph aureus bacteremia were randomized to receive background treatment, so a glycopeptide or a penicillin, depending on whether it's an MRSA or MSSA. But the randomization was based on whether they got rifampicin on top or not. So this was a randomized trial that tested combination therapy for Staph aureus bacteremia versus monotherapy. Only a small proportion of patients had MRSA, but that's a different discussion. The first thing you will see is that the primary outcome, which is having microbiologically confirmed failure or recurrence of the infection, was not different. So in other words, all your colleagues who were not given rifampicin and you were not able to sleep at night because your patient is on rifampicin, it turns out actually that didn't matter, because rifampicin didn't make a difference to those patients anyway in terms of outcomes that mattered. Even worse, rifampicin was associated with more frequent discontinuation because of toxicity and because of drug-drug interactions. So here's something that you might want to consider. Will you change your practice and stop giving rifampicin or a second agent when you're treating Staph aureus bacteremia? I did. I don't give rifampicin or anything else. Newer agents are always attractive. And it's always a good idea to say, well, what if I have a new agent? Would that change my mind? Would that change what I would do? ERADICATE, which again was discussed this morning, I think more than once, is a very interesting study. It discusses a very interesting new agent, an anti-MRSA cephalosporin, again, something that I would not have expected myself to utter only a few years ago, because we were always told that MRSA was resistant to all beta-lactams. Turns out this was wrong, and we had to relearn that. So ceftobiprole is an anti-MRSA cephalosporin. It has activity because it's able to avoid MACA genes and target. In this randomized trial, patients with complicated Staph aureus bacteremia were randomized to receive ceftobiprole or daptomycin. I skipped daptomycin because it never changed my practice, so there was no point discussing it. But when those two agents were compared in treating Staph aureus bacteremia, essentially ceftobiprole was non-inferior, which is the primary endpoint and how this study was designed. If you're a purist, if you're pedantic, you'd say, okay, it's non-inferior, but is it statistically superior? And the answer is no, of course, because the confidence interval crosses 1. So there is no statistically significant difference between them, but it's also non-inferior. Now, if you were to introduce a new agent and then ask yourself, should I be using it instead of a previous agent? The first question you'd ask is it better? Now, what do you mean by better? Well, it could be better in efficacy. The answer here is, well, really, no. They're equivalent, okay, non-inferior. The second reason to switch is maybe it's safer, and the answer is actually not. It's not safe. So if you look at the incidence of adverse events in ceftobiprole compared with daptomycin, and you know that daptomycin is not a safe drug to begin with. I mean, it failed to show better tolerance compared with vancomycin and some of the older stuff. So this is an agent that is useful, of course. But for me, it's not something that I would suddenly run to. It's not something that would change my practice. In Staph aureus, 25 years ago, if you have Staph aureus bacteremia, a patient with Staph aureus bacteremia, and it's an MSSA, you would give them a penicillin, Gluclox, or you would give them Cefazole. If it's an MRSA, you would use a glycopeptide. 25 years later, you would do exactly the same. Now, if I showed you more and more studies, but Raheem did a fantastic job of doing this morning on treatments and outcomes of Staph aureus, you would see antibiotics were introduced in the 1940s. Mortality due to Staph aureus bacteremia in the 1940s was 30%. Can you guess what is the mortality with Staph aureus bacteremia today? It's still 30%, despite vancomycin and despite Fluclox. So where we started, unfortunately, is where we are now. The only thing that I changed in my own practice, and you can agree or disagree with me, is that I now think carefully before I expose my patients to unnecessary, potentially additionally toxic agents. And I just stick to the rules. And I know that it's not a very nice emperor, but it is the emperor, and it continues to reign. Glycopeptides or anti-staphylococcal penicillins, cephalosporins. And if you know any better ideas, then please offer them to your patients. The next one that changed my practice was Merino trial. And this would be much more familiar, I'm sure, with the audience because it's relatively recent. So even the youngest among us, you might be aware of that. So a few years ago, more than a few years ago, I should say, maybe a decade ago, we had these interesting discussions about whether it would be okay to use piperacillin tazobactam to treat ASBL-producing Enterobacterales bacteremia as long as it looked susceptible in vitro. And we had this interesting debate clinically. And then we had a run of observational studies that didn't agree. So some studies, typically from America, that say, yeah, looks like it works. And then studies typically from Europe say, no, looks like carbapenems are better. We never knew the answer. Merino was a randomized trial that was specifically designed to answer this question. So they randomized patients with cephalosporin-resistant Enterobacterales infections, mostly because of ESBL producers, and they randomized them to receive meropenem or beptazo. In vitro, the patient must have an isolate that is in vitro susceptible. So they randomized them, and the primary endpoint was mortality. And as you all know, it turns out that actually beptazo is not as good as meropenem. And it turned out that a lot more patients were dying in the beptazo arm compared with the meropenem arm when they were used to treat patients with cephalosporin-resistant Enterobacterales infection. Now, the first thing that comes to your mind is, but why is that? Well, I don't know why, but this is what happened. Subsequent work tried to explore more about those isolates and why there was this excess death in the beptazo arm. And one obvious possible explanation is that simply the labs where testing was done just got it wrong. So if you were a recruiting center and you had a patient with Klebsiella bacteremia and your lab reported it as susceptible to beptazo, well, maybe it wasn't. So one very good thing about Merino trial is that they included that the recruiting centers sent their bacterial isolate to a central lab down in Brisbane in Australia for reference lab testing. And what you're looking at now is a follow-up publication on Merino that describes the MICs of those isolates as tested in the reference laboratory. So now the question of, was it because of resistance or not, could be clearly disentangled. And what they show, the first thing is, of course, diagnostic labs are diagnostic labs, so they're not going to have the same accuracy as a reference lab. So some isolates were indeed misclassified. So although they were reported as susceptible, they were actually resistant. But more worryingly, I'll skip this one, more worryingly is that when you look at the MICs, now from the reference lab, you see that even isolates that had low MICs were still dying. So in other words, when it looks like Bibtazo is just not as good as carbapenems for treating ESBL-producing organisms, at least if you're trying to treat bacteremia. And I had to change my practice because I used to use Bibtazo for these organisms as long as they're reported susceptible. But I couldn't do that anymore. So Bibtazo no longer for ESBL-producing Enterobacteriales was another practice-changing trial for me. Part of what I do, Nizar said this in his introduction, is that I look after patients who are immune compromised, especially hemato-oncology patients. And as you know, a lot of those patients experience neutropenia, and at least half of them will have fever at some point. And when I was a very young man, I was told that if you have a patient with febrile neutropenia, then before you eat or drink or breathe, you start them on antibiotics. And I said, yes, sir. What should I start them on? And it was always an anti-pseudomonal, preferably, well, sometimes in combination, sometimes not, but that's a different discussion. So it was ingrained in my very small ID brain that I have to give antibiotics and I have to give them very quickly. But no one took me aside and told me what should I do next? As in, when do I stop them? Started them, but what do I do about stopping them? And what became common wisdom is that you just carry on and on and on until the ANC recovers, even if the patient is afebrile. Now, that was okay in the 1980s and the 1990s, but come on board ESBL producers and carbapenem resistance, and that became, ooh, we're now seeing extremely drug-resistant bugs in patients who are difficult to treat to begin with. Surely we need to think about how long we need to treat. The Europeans took an initiative, and they started investigating whether it would be okay to stop antibiotics. Let me start from the beginning. So you have neutropenic fever of unexplained origin. You give empiric antibiotics, antibacterials. The patient becomes afebrile, clinically stable. There's no focus of infection, but they're still neutropenic. Should you stop antibiotics? The American guidelines, IDSA guidelines, which were last updated in 2011, say no. You continue antibiotics until the ANC recovers. So the Europeans started to test the water and said, no. If they're well and stable, we'll stop. So the first trial, which is the one I'm sharing with you now, is called How Long? Very aptly named. And the question is, how long should you continue once you've started? So this study randomized patients with febrile neutropenia who became afebrile and are clinically stable and don't have a focus of infection, have been afebrile for at least 4 days, to either continuing antibiotics, on and on and on and on and on, or to stop. And the primary endpoint was whether they died or not. And the answer is A, they obviously received less antibiotics if you stop the antibiotics. B, they actually were not more likely to die at all. And very interestingly, they actually had less fever. So this was a trial that was able to demonstrate that stopping empiric antibacterial therapy in patients who have febrile neutropenia once they are afebrile and don't have a focus of infection is okay and is safe. And it allowed me to change my practice. This was published, but alongside it, soon after or before, many other cohorts from different parts of the world were also testing the same. These are cohort studies, so they don't meet the criteria that I set myself at the beginning. But what happened is many cohorts came along, eventually one cohort from the United States. And if you look back at the guidelines, you find that the European Conference in Treatment of Leukemia back in 2013 said, yes, we should be able to stop. Took other individual European countries a little longer to bring it on. And the MD Anderson guidelines also say you can. I can't tell you what the IDSA guidelines say because the last version I have is 2011. And I changed my practice a long time ago. So I started at a point where everyone was receiving empiric antibacterial therapy forever. And now we don't. Now, if my patient is afebrile and is clinically stable and doesn't have a focus of infection, I stop after 4 days and I just go back to the basics. And if they develop a new fever, I treat all over again. Is this likely to kill them? The answer is no. And I can say no because I can point at the results of the How Long trial. I think this is the last one. It's also a favorite of mine. Again, in the context of neutropenia, but this time persistent febrile neutropenia. Patients who continue to be febrile without an explanation. Again, in the 1970s, early 1980s, wisdom had it that you needed to add an antifungal. Pizzo and his colleagues published work that resulted in this becoming the standard of care. And on it went. Everyone who had persistent febrile neutropenia was slapped on antifungals until there's no tomorrow. The reason this was done at the time was that the diagnostics were very limited. And if you had patients with unexplained fever, there was no way that you could tell whether they did have invasive fungal disease or not. But in the last 20 years or so, we have better diagnostics. So we can use biomarkers and we can use imaging. So again, it became more sensible and pressing to ask the question, do all these patients really need antifungal therapy, or can we be more selective? It started initially treading very carefully, feasibility studies, small studies, but in come this important study from Melbourne in Australia, multi-center in Australia, but it was led by Melbourne Center. What the investigators did in this trial is they randomized patients who were high-risk hematology patients, including allogeneic bone marrow transplant recipients and those who are receiving chemotherapy for acute leukemia. And they were randomized to one of two pathways. One pathway is fever, give them antifungals, fine. The other arm had a diagnostic strategy. And that diagnostic strategy included doing galactomannan and panfungal PCR once a week. And if the patient developed the fever, they would do the biomarkers, the PCR and CT scan and clinical evaluation. If they had evidence to suggest invasive fungal disease, they'd receive an antifungal, otherwise not. So just to take you back a couple of steps, this means withholding antifungals from some patients who were febrile but don't have any additional evidence of invasive fungal disease. The primary or the outcomes that they looked at were use of antifungals, obviously less in the diagnostic arm, but very interestingly, there was no difference in mortality. Your patient is not more likely to die because you didn't give them an antifungal, despite their persistent febrile neutropenia, if, that's a very important if you have a diagnostic pathway in place that can help you identify them. And you also diagnosed more invasive aspergillosis. Now, the reason I call this fool's gold is 50%, I want you to process this number, 50% of the patients in the diagnostic-driven arm who were diagnosed with invasive fungal disease did not have fever. Fever is a fool's gold. If you're chasing fever to diagnose invasive fungal disease, you're actually missing a lot of patients. If you're giving antifungals because of fever, you're actually giving antifungals to a lot of patients who do not need it. So we started at a point where we were misfiring antifungal therapies all the way, and then a randomized trial was able to change at least my practice and allow me to be a little bit more selective. And by using diagnostic-driven strategies, I can confidently and safely withhold treatment, at least for some patients, and make sure that others are treated as early as possible. Now, I stood here for, I think, about 30 minutes talking to you about trials that changed my mind. And as I said to you at the beginning, these may have changed your practice, may not. Maybe there are other things that you see are far more important and were more worthy of those 30 minutes than I did. But that's a difference in perspective, I guess. But what I want to finish with is Alvin Toffler. This guy is dead now. But Alvin Toffler is what you call a futurist. So he's someone who analyzes history, he studies history, he studies present, and tries to predict what happens in the future. His first breakthrough book was published in 1970, and it was called The Third Wave. And in it, he predicts things like there will come a day when kids will be running around with a tablet, essentially an iPad. That was written in 1970. His book that was published in 1990 was Power Shift. It was his last book. And in Power Shift, he talks about three forms of power: force, physical force, wealth, and information or knowledge. And he says that knowledge is the most powerful of all powers. Because if I have more knowledge, if I have more information, I would know a little bit more than you. But also Alvin Toffler says in the same book, we live in the age of information. We had the two revolutions in the past. So we had agriculture and then industrial revolution, and now we're living in the information. But he says that to be alive in the 21st century now, he says that unless you are able to learn and then unlearn and relearn again, you won't be able to go much further. In other words, I spent the last 25 years doing infectious diseases, and God knows how much before that doing other things. And obviously, I've accumulated a lot of stuff. Learn. But there comes a point where unless you consciously unlearn and offload some of what turns out to be inappropriate, incorrect, no longer really relevant, you won't be able to learn again. And it's only if you're able to combine those three that you can move forward. So in 25 years, that's all really I learned. And what I learned today is very, very simple. I actually know far less than I think. And every day I discover more that I do not know. And I still haven't learned enough. Thank you.